Hello everybody, my name's Susan Deuchars and I'm a neuroscientist at the University of Leeds. And in this presentation, I really want to make you aware of how important and exciting scientific research is, and science as a whole is. There are some really great quotes out from people who are scientists, and I've just put this one up here because I thought it was fantastic. Fabiola Gianotti is an Italian physicist, and she's here in front of the machinery that is part of her everyday life. But her quote is that this job is a great scientific adventure, but it's also a great human adventure. We have made giant steps forward. However, what we know is very little compared to what we still have to know. And that is so true of scientific research, whether you're thinking about medical research, whether you're thinking about physiological research, whether you're thinking about plant research or climate change, researching into climate change. We know an awful lot more now in this time than we did previously, but it's very clear that we've still got a lot to understand. And that's why scientists are passionate about trying to make a difference. We really care about how our research may help others, help our planet, help plants to grow properly, help animals to survive. It's all about trying to make the world a better place, and that's what really drives scientific research and science in general. I also wanted to show you this quote from Mae Jemison, who was one of the first female astronauts to go into space and the first female person of colour to go into space. And I love this quote because it's so true. Some of the most fun people I know are scientists, and they are. Being with scientists, learning from them, laughing with them, listening to the problems that you may have at times, and celebrating success. It makes it a really fascinating and very, very rewarding job to go into. So what I thought I'd do is to let some scientists, some female scientists, speak for themselves today, and telling you what they love about their science, some in slides and some in their own words. So I hope that you'll enjoy listening to them. I thought I'd start by telling you a little about my scientific research and why I study what I do. I'm a neuroscientist, and therefore I look at how our brain cells, our neurons as they're known, actually function in our bodies. We have brain cells or neurons in our brain, obviously, but also in our spinal cord, and also out in the nerves, which are going out to innervate all of our muscles. We've got them within our gut, actually controlling our gut, for example, and all organs. They're critical, and we need them to function properly. But what happens when they go wrong? You have conditions where neurons, brain cells, are lost. And you have other conditions, such as multiple sclerosis, where actually the signals that neurons actually rely on to be able to pass information from our brains all the way down our spinal cord and out to our muscles, where that signal is slowed. This signal is actually an electrical signal, as many of you may know. And this signal, or action potential as it's known, travels very, very rapidly down the axon of the neurons to actually release neurotransmitter at the base of the neuron. And this relies on having this myelin sheath, which is around the axon, to enable the signal to travel very rapidly. And that myelin is produced either by Schwann cells out in the peripheral nervous system, or these specialized cells called oligodendrocytes in your central nervous system. Now, in multiple sclerosis, what happens is your own body actually attacks that myelin sheath, and the myelin sheath is lost around the axons. So that means that the signal, the electrical signal, the action potential, can't travel as rapidly down the axon, and therefore it affects people's ability to speak, to think, their vision, down in their spinal cord as well, their senses, actually feeling things within their body, their muscles, they don't work as well. They have problems with their urinary system, so it's more difficult for them to actually go to the toilet, and in their digestive system as well. So you can imagine that these conditions that involve loss of cells in the body really are quite profoundly affecting a person's life, a person's ability to carry out their normal responses. So we need to do something about that. Now, what I've been studying, and I have such a fantastic group of scientists around me working with me, are looking at whether or not we can replace some of these cells. Now, a way in which we can do this are by using replacement of neural stem cells. So if you look at your spinal cord down here, these are the vertebrae, this is your backbone as you talk about. So these are your vertebrae down here. And if you cut through your vertebrae and your spinal cord, you can see that your spinal cord looks a little like this tiny, tiny thing here, protected very beautifully by the vertebrae. If you look across at that cross section, right in the center of the spinal cord is the central canal. And actually, it's not just a boring little circle in the middle. There are masses of cells in this region which are considered the neural stem cells of the spinal cord. So what do we mean by neural stem cells? Well, basically, that's what we need when we're first developing as babies. We have many, many millions of neural stem cells which are reproducing, becoming more and more numerous in number, and then they start to differentiate and they become either neurons, which are very important, or astrocytes, which are really important supporting cells, or these oligodendrocytes, which actually form the myelin sheath. And so we're interested in looking at whether or not we can make our neural stem cells within our bodies become oligodendrocytes and actually replace that lost myelin. And I'm very lucky to be supported by some fantastic researchers, people like Nur Hagani, who you're going to learn about in the talk later on, Nasla Shafin, Barinder Lal, and others that I'll talk about during the talk. So we want to increase the numbers of new cells, of these neural stem cells. So if you consider that these are the neural stem cells here, we want to try and help in a condition like multiple sclerosis. So we want to drive these neural stem cells to replicate, to get more of them, so they're proliferating, which is what we call it here. And once they've proliferated, we want to drive them to become immature oligodendrocytes, and then over time mature to become fully functioning oligodendrocytes that can replace that myelin sheath we've lost and get repair. And this is what we do in our lab. So we add a drug to boost the production of these stem cells. We can get more stem cells if we take cultures of spinal cord, for example. So you can see these are more and more stem cells being produced in the central canal region here. And we can also use models of conditions such as multiple sclerosis, where we're losing the myelin sheath, just as what happens in our own bodies with somebody with multiple sclerosis. And we can look to see whether if we lose that myelin sheath, we can produce more stem cells to produce more oligodendrocytes to repair the area. And that's what these green cells are here. This is the damaged area. And we're actually producing many, many more of these cells which can replace the lost myelin. And you can actually cut through this myelin and look to see whether we've replaced it. And this is what we're showing here. So this is the myelin that we have around our cells here, our axons. And if you look at it in the machine that we were talking about, the electron microscope at very, very high magnifications, we can look to see whether our treatment increases the amount of myelin around the axon. So this is cut through the axon like that. And we're looking at that myelin, and that's with no treatment. You can see that the myelin is very, very thin, this black line. But if we treat with our drugs, then we actually increase that myelin sheath, and we can actually help to remyelinate the axons and help with recovery. So that's why I'm passionate about science, because I feel it's really, really worthwhile. And as you can see, these are the people who are also passionate about science: Lauren and Claudia and Laura and Jill and Lucy and Nora. They care about trying to make a difference to help people with these conditions. There are other fantastic scientists who I work with. So within Leeds, we've got 3 incredibly talented scientists who are looking into replacement of other types of tissue. So we have organ damage, we have tissue damage that occurs as we age or in certain conditions. And so these brilliant scientists are actually looking at ways in which they can replace that damaged tissue or that damaged organ. So the first one I'd like to tell you about is Dr. Helen Berry. And she's interested in, so she's really driven, she says she's driven by curiosity and a passion for science applied to medical needs, which is exactly what she's doing here. So we know that in many heart valve diseases, we get degeneration and dysfunction of the heart valves, which are so important. These valves here, which are within your heart, so this is a picture of your heart here, these aortic valve, mitral valve, pulmonary valve and tricuspid valve are absolutely critical in making your heart function properly. But over time or due to disease, you may get dysfunction, of these heart valves. And so what you need to do is actually replace these heart valves, actually replace them with something which is synthetic, which is or decellularized, that will actually help in these conditions. And many children are actually born with defects in their heart and blood vessels, for example. And there are many, many people in adulthood who suffer from congenital heart disease, which is what we're talking about here. So they need multiple surgical replacements. So we need to repair and regenerate this tissue. And that's exactly what Helen looks into. So this is what she's thinking about. Tissue engineered valves, making new valves which are actually accepted by our bodies and will enable recovery of function. So they need to have growth and regenerative capacity. And therefore, if you do this, and what she's interested in doing is using these decellularized donor valves. And it seems as if these are working so well because now Helen's got over 10 years of clinical data from Brazil and Europe to look at how this works. And so what we need to think about is actually making a better performance. And this is what is happening at the moment. This clinical translation to other locations as well, and making a difference. 2 other scientists I'd like to let speak for themselves because they've actually annotated and spoken over their slides to enable us to understand what they're doing. Dr. Hazel Fermor and Dr. Jen Edwards will also look at ways of replacing worn tissue or worn organs. And so I'd like to let them speak for themselves.
Hi, I'm Hazel, and I really enjoy being a research scientist because it gives me the opportunity not only to work with a wide variety of really interesting and intelligent and exciting people, but it also gives me a bit of a purpose. I know that what I'm doing in my research lab is leading to treatments for people who are suffering, and I can improve people's quality of life. So I lead research in my group into the treatment of osteoarthritis, which is degeneration of our synovial joints, such as your hips and knees. And this affects a huge number of people worldwide, and especially in the UK, there is around 9 million people that suffer with pain and loss of mobility in their joints. What we're doing is developing early intervention regenerative therapies, so in the form of decellularized scaffolds and also injectable hydrogels to restore and regenerate functional tissues such as cartilage and bone in joints to prevent the progression and development of osteoarthritis. We also do a lot of work looking at how we can preclinically assess whether these treatments actually work, which I'm happy to say we are in the process of doing for bone and cartilage scaffolds. And we also work really closely with our clinical and industry partners to make sure that we're on the right track in terms of getting products that you can actually manufacture at an industrial scale, and that our clinicians and patients are actually asking for the products that we're developing, so that we've got a much better chance of actually being able to help people at the end of it, which is exactly what I got into this job to do.
Hi everyone, I'm Jen and I do research into healthy and active ageing. The reason I enjoy my work so much is that it feeds my curiosity. I get to work with a wide range of people who are just as curious as I am and apply my biological and engineering skills so that we can better understand the tissues in our bodies and how we can repair them. I'm working on healthy and active ageing by repairing musculoskeletal injuries, and my particular focus is in the foot and ankle. I use decellularization, which is a washing process where we remove the cells from natural tissues, but we leave the extracellular matrix behind. And this means we've removed the cells which cause an immune rejection, but we have a tissue which is strong enough to perform as a replacement in the body. One of my interests in particular is in adipose, and we might think of adipose or fat as generally a bad thing, but it's particularly important in some areas where it has a cushioning role, such as in the sole of your feet. There's a clinical need to prevent the formation of diabetic foot ulcers, and these form on the sole of the feet of diabetic patients, often related to degradation of the plantar fat pads. Although these ulcers are small, they have a huge impact. Treating them and their associated complications costs the UK health service over £1 billion a year. But perhaps more importantly than that, the mortality rate of someone who has healed one of these ulcers at 5 years after the ulcer is higher than that for breast cancer or prostate cancer. So they have a really big impact in patient quality of life and also whether or not you're going to survive very long. So I've been working with a local surgeon and a research and development team within the UK Health Service to develop a decellularized adipose. So far we've produced it in a sheet form, but we're working to turn it into an injectable hydrogel so that we can restore the fat pad, prevent these ulcers, and hopefully save lives. And all of this helps me feed my curiosity, but applying my knowledge to a real clinical challenge with the potential to help many patients around the globe.
Thanks, Jen. That was really fascinating. And it's lovely to learn about Jen's work and Hazel's work and why they
Hello,
care.
everyone. I'm Jessica.
So I'd like to move a little bit to Jessica. And Dr. Jessica Kwok is actually a neuroscientist like myself. And she's also interested in repairing or helping repair. So let's go back and listen to Jessica.
Hello, everyone. I'm Jessica Kwok. I'm an associate professor at the University of Leeds. So what I'm going to tell you in the next few slides are about my research and how I arrived there. So I'm originally from Hong Kong. I did my undergraduate degrees and postgraduate degrees from the University of Hong Kong, and afterwards I moved to the UK, and now I'm teaching at the University of Leeds, and at the same time I'm also doing research in my lab, mainly focusing on enhancing neuroplasticity for regeneration. So what is neuroplasticity? Neuroplasticity is actually a word to describe the ability of the brain to adapt and change and to adapt to the external environment. And this is really important when we have diseases in the brain, such as after spinal cord injury, when we have a disconnection in our neuronal network, or in order to enhance memory in aging, for example, or also in dementia as well. So how do we achieve that though? So you might have heard about this structure called synapse. Synapse is the basic talking unit in our nervous system. They communicate between different cells. And in order to achieve or to increase neuroplasticity, what we are focusing in the lab is a structure called perineuronal nets, mainly made with sugar on the surface of neuron, and they are the one actually wrapping around the synapses, consolidating synaptic formation. So if you look at perineuronal nets on the surface of a neuron, that is how it looks like. It is wrapping the surface with holes where the synapses are formed. So in order to enhance neuroplasticity, what we need to do is to remove perineuronal nets. A situation very similar to connecting electrical cable. You need to remove the electrical cable, remove the plastic sheath on the electrical cable, making new connections, and then reseal it, allowing the system to function properly. And by regulating perineuronal nets, we can actually modulate quite a number of different pathologies in the central nervous system. What I'm focusing in my lab is in spinal cord injury to try to enhance functional recovery. And also we are trying to enhance memory, particularly, as I said earlier, in aging and Alzheimer's disease model. But perineuronal nets is also involved in a lot of different diseases. And by understanding perineuronal nets and by designing tools in modulating perineuronal nets, we will be able to provide some improvements in all these diseases you are seeing here. So the potential of our research is really big because it can improve a lot of different diseases. It sounds like really grand, but of course I didn't set out my research career as to actually improve all these conditions. So what I really want you to understand is that in order to achieve big goals in our life, what we will need to do is actually taking them step by step, setting small steps at the beginning in order to allow us to achieve the big goal at the end. So you might also think, okay, well, some of the steps may actually be too big and you cannot actually reach there and you're afraid of failure. I think that is something that we need to take out of from our mind. If you fail in one step, what you need to do is actually setting smaller steps. Instead of having the big one that you set originally, just reduce your expectation slightly and try to achieve them one by one gradually, and you will eventually actually reach the top. So a key message which I want you to understand is to take small steps in order to reach the big goal. So I hope you enjoyed the talk. Thank you very much.
I really enjoy listening to Jessica always when she talks about her work because she's so passionate about it, about making a difference. And you'll also realise the passion comes through with our next speaker, who's Dr. Beatrice Felipe, who is originally from Italy, but she works on how we can think about controlling our blood sugar and our weight. And she's really interested in the brain circuits that actually control our insulin levels and our blood sugar and our weight. So let's listen to Bea telling us about her research.
Hello everyone, my name is Beatrice and I am an associate professor at the University of Leeds. Today I would like to tell you a little bit what we do in our laboratory. In particular, we are interested in understanding how insulin, that is an important hormone that decreases blood sugar level, can also signal to the brain to regulate metabolic function. So what do we know is that this area of the brain, highlighted in yellow, called the dorsal vagal complex, can sense changes in insulin level and decrease food intake, body weight, and fat deposition. In addition, it can also decrease blood glucose level by decreasing the amount of glucose that is released by the liver. What is very interesting to us is that if we feed our rodent model, our rat model, with high-fat diet to make them obese, they become insulin resistant. It means that insulin cannot anymore signal to the DVC and cannot anymore control our metabolic function. This work was done by Bianca Patel, that was a PhD student in my lab. In addition to that, we also know that the dorsal vagal complex can control thermogenesis, and with it also the way in which we burn calorie. And this work is done by Arianna Fozzato, a PhD student in my lab. We also are interested in understanding which neuronal network, which neuron in the DVC can sense insulin to control metabolic function. And Lauren is carrying out this project. We also want to understand at the molecular level which molecules are altered by high-fat diet feeding, and Jessica is trying to understand and answer this question. Finally, we also look at insulin sensing in that area of the brain, and in particular, we are interested to see how the olfactory system can sense insulin to regulate metabolism. So, to conclude, our main aim is to really understand how the brain can respond to changing in hormonal level to regulate our energy homeostasis.
Thanks, Bea. As you can see, her enthusiasm really comes through. She loves talking about her research, and this team of very talented young scientists are really making a difference in our understanding of how these brain circuits work. It's such an exciting area of research to be in. What about things like personalised medicine? Because there's a real push at the moment to think about how we need to actually target our medical therapies to people depending on their situation, depending on their condition, depending partly on their genetics and what the mutations might be. So I'd like to hand over to Dr. Natalia Roba Delgado, who will talk you through her research into personalised medicine. So over to you, Natty.
Hello, my name is Natalia Riobo Delgado, and I am an associate professor at the University of Leeds. I work in the School of Molecular and Cellular Biology, and in my position I teach and I do research, and my interest is to study hedgehog signaling to improve personalized medicine. My journey to the University of Leeds was not direct. I was born and raised in Argentina, and I did my undergraduate degree in biological sciences and my PhD at the University of Buenos Aires in the exact and natural sciences faculty. From there, I moved to Philadelphia in the United States, where I did postdoctoral training at the University of Pennsylvania. And then I started my first faculty position at Thomas Jefferson University, where I started to run my independent research group. After several years at Jefferson, I moved to the University of Leeds, where I am currently now. I am interested in studying hedgehog signaling because there is a plethora of diseases that are caused by excessive or deficient hedgehog protein signaling. For example, during embryonic development, defective hedgehog signaling can cause holoprosencephaly, which is a rainbow of congenital defects that prevent proper separation of the brain hemispheres and of facial features, including the eyes in the most severe cases, causing cyclopia, the nose, nostrils, and the lip. There is also diseases for excessive hedgehog signaling in embryonic development that causes polysyndactyly, numerous fingers and toes that sometimes they are fused together. In adults, excessive hedgehog signaling in the skin can cause tissue fibrosis, for example in the disease scleroderma, in which increased collagen matrix deposition by fibroblasts in your skin causes the skin to harden, and it causes real disabilities in these patients that, for example, cannot use their hands, sometimes requiring a hand transplant. They also present with lung fibrosis, which really impedes their ability to breathe normally, and as you can imagine, is a very terrible disease. Excessive hedgehog signaling also causes cancer. For example, mutations in signaling pathway components can cause cerebellar cancer in children, which is called medulloblastoma. It's also the cause of the most common cancers of all, which is a skin cancer type called BCC, of basal cell carcinoma of the skin. And it also causes or is involved in the progression of many cancer types, including colon cancer, breast cancer, prostate, and lung cancers. Therefore, studying hedgehog signaling, I'm trying to understand what is exactly the defect in each of these different conditions to try to develop effective targeted therapies.
Thanks very much, Natty, for taking us through that. It's really interesting to hear how Natalia has moved around and enjoyed her science and how much it means to her. you can see that actually looking at some of these conditions is really, really important in helping to either stop these problems in the embryo or in adulthood, or indeed to try and treat cancers. And that's where personalised medicine is important. But we also need to think about other types of science. And there are some fantastic scientists who are looking at things like crop management and looking after our ecosystems. So I'd like to tell you a little bit about two different scientists. Jocelyn will speak for herself first, and then I'll show you some slides from Maria Bega. And they're really looking at very different aspects of science, which are absolutely critical in the world today. Because if we don't have good crop management and a control on our climate, then we will not have a world in which we will be living and worrying about our different conditions or diseases or cancers. So let's listen first of all to Jocelyn.
Hello, my name is Jocelyn Benitez Alfonso, and I am a plant scientist and associate professor at the University of Leeds. I work in understanding how plant organs, shoots and roots communicate, and how this communication determines plant responses to changes in the environment. I study small microchannels that are named plasmodesmata. They serve as a route for the molecular transport of signaling proteins, RNAs, and sugars. And also, they serve as gateway for the spreading of viruses, among other diseases. My most recent project investigates the structure of these channels and dissect the properties of their components. The aim is to identify new knowledge that allows to improve plant growth and to exploit the properties of plant polymers in the design of new materials, ideally from sustainable sources such as agricultural waste. These outcomes will make an impact on the crop biotechnology and biomaterial industry, addressing global needs to improve food security and to reduce waste. But I was not always a plant scientist. I was born in Cuba and studied part of my degree in chemistry at the University of La Habana. After my second year, I moved to Europe, to Spain specifically, and to the University of Córdoba, where I continued my studies in biochemistry. I did postdoctoral research at Cold Spring Harbor Laboratories and at the Young Innes Center in Norwich, where then I was introduced to plasmodesmata and plant science before identifying this interdisciplinary area of research and established myself at the University of Leeds. What I have learned through this journey and studying plasmodesmata is the power of communication and collaboration, which has been critical to drive my interdisciplinary career. I believe that by embracing diversity, as a community, we can face great challenges, and that is my message for you today. Thank you for your attention, and here you can find my email or contact me via Twitter at Benitez Lab. Thank you.
Thanks so much, Jocelyn. I think it's very clear from Jocelyn how the importance of collaboration, and she also spoke about interdisciplinarity, learning from each other. So listening to somebody like Jocelyn and actually taking her information and actually applying it to our own science. So learning from other scientists is absolutely critical. And as Jocelyn mentions, collaboration is really, really important. So the last person that I'd like to tell you about is Maria Bega, who has done some really beautiful research into climate change and really making a difference at the level of the climate and in looking at kelp forests and coral reefs. And what I loved about Maria was she provided this quote for me. So she says, I love discovery and I'm a sucker for trying to solve unsolvable challenges, sometimes to my own detriment. She says it's really quite difficult at times. I am also very passionate about having purpose, and my research helps to save coral reefs and enabling people who depend on them to maintain their livelihoods and enjoyment of these ecosystems. She loves teaching students and exciting them about making a difference or simply just being thoughtful and smart human beings. And I thought that quote was absolutely wonderful to be able to put in, because this is straight from Maria. She sent this to me. And it's about what I love with science is thinking about being a thoughtful and caring about the environment in her research, or others, or plants, whatever it is, but being a smart human being. And whether you become a scientist for the rest of your life, or whether you use your science to actually make a difference, that's what it's all about. And just to finish, I'd like to show you a couple of Maria's slides, if I can. So this is her research. She looks at ecological understandings. Sorry, her slides are a bit slow in moving. So she's got a lot of figures on here. So you can see she's actually looking from the level of actually collecting data from specific species to look at the abundance or biomass of these species. She looks at the traits of these species over time, so and looks at climate models alongside it. So she considers these environmental parameters to think about the model responses and what's going on, and therefore to try and manage the solutions overall. So thinking about developing conservation solutions. So her work is really and truly making a difference. And she talks about tropicalization research, so all about trying to predict the reorganization from tropical to temperate climates. So these are actually pictures of the same area, and this is a temperate kelp reef in 1985. And over time, by 2013, it had become this coral-dominated reef. So the tropicalization, this is exactly what she means, occurs in a very, very short period of time, from 1985 to 2013. So you can see that the reason why Maria is so passionate about her work is looking to see what is happening in our seas and in our reefs to see whether we can actually stop that degeneration of these beautiful areas. And so the overall message that I'd like to give you today is, no matter what area of research you're interested in, no matter what area of science you're interested in, you'll find that you'll become incredibly immersed in it, incredibly passionate about what you do. And I'm hoping that that has come across in this talk that I've given today, and that you find something along some of the research areas that I've covered to really inspire you to consider studying science at degree level or at A level, whatever you feel that you'd like to do. So thank you for your attention, and I really hope that you will become a lover of science just as we all are.